This is your Discovery Lab lecture for Wednesday, March 24th. Today we're going to be talking about drug discovery and the process of drug development leading up to clinical trials. Here's an overview of what we're going to be talking about. As usual, we'll go over the assignments and the upcoming schedule, both in lecture and in lab. I'll give you a very brief overview of what's going to be happening in lab in the next couple of weeks, though for details you should be watching the pre-lab video every week. We'll back up and talk about what we've already covered before moving forward to drug development. And I'll give you a little preview of what we're going to be talking about in the coming weeks, which is clinical trials and bioethics. We are here in March 24th. And leading up to this week, you would have completed one of the two optional drug development assignments on either Perusall or PlayPosit. These weren't graded assignments because, as you know, this is the academic pause week, so we don't really have any formal grades this week. Next week, we're going to be talking about clinical trials, so your PlayPosit for the clinical trials unit is due on March 28th at 10 p.m. as usual. So we'll be back on our normal Sunday assignments due at 10 p.m. schedule. Sunday, April 4th is when your methods section is due. That was moved from March 28th to give you a little bit more time to think about the methods that you'll be reporting on your poster. And we'll be talking about bioethics and reliable science that week in lecture. This is our last content week for lecture. And then after that, we'll have two more lectures, I think, that focus on the Cure Symposium and the assignment that's due during finals. Let's take a quick look at what's happening in the lab. Just to review, our experiment is based on fly development. You collect embryos or eggs in the population cages, allow them to develop into third instar larvae, and then we treat those third instar larvae with compounds in the food, we irradiate them, and then evaluate about 12 days later whether or not the larvae develop into adult flies. You'll be doing that this coming week for most of your vials, so you'll be using a light box and a marker to keep track of which pupil cases have a closed and which haven't. So get used to using the proper terminology. I hear people saying cocoons and hatching. Those names aren't necessarily wrong, but in Drosophila research, we're going to use the terms pupil case or pupae and a closing, which means emerging from the pupil case. The very first experiment that you did was a high-dose experiment, and this experiment was designed to test the highest achievable dose in someone's serum. The idea is to see whether it works in the absolute best case. The second experiment aimed to determine what's called an MIC, or the median inhibitory concentration, so the concentration at which 50% of the effect is observed. This experiment also allows us to determine whether the compound that you're studying is a poison that we can't control or a drug that we can control. In the next week's experiments, the week of March 28th, I think it would be interesting to take your compounds and repeat that dosing series with or without radiation. So if you can imagine taking a look at the effect of your compound, it has some ability to kill the cells, probably. If the compound is working with radiation, we might not know whether that effect is an additive effect or a synergistic effect. In other words, when you put drug and radiation on the larvae, are you just seeing a simple addition of the effect of radiation plus the effect of the chemotherapy, or are you seeing something more than that, where the chemotherapy or your compound is actually sensitizing the cells to be more responsive to radiation? And that's called a synergistic effect. So if you choose to do something like this, where you're testing the compound by itself and testing radiation by itself and then testing the two together, we can then figure out if the drug, say the drug has the ability to kill 30%, we'll just call it 30% of the larvae. And then say the radiation by itself kills 50% of the larvae. If you add the two together and the effect was additive, you would have 80% death or only 20% survival. If it were a synergistic effect where the two are somehow working together, you would have greater than 80% death, bringing it down probably close to 0% survival. So that experiment will help you understand whether your drug and radiation are acting additively or synergistically. Next week will be the last week of testing, so March 28th. During those two labs, you won't be doing any testing beyond that week. So this is your last opportunity to set up vials, and then in the next 2 weeks, you'll be evaluating survival and developing your poster and your presentation of your poster in the lab. Now, before we move into this week's content, let's back up a little bit and take a look at what we've covered so far. So you're doing cancer research, and one of the nice things about doing cancer research is you don't really have to pitch it that strongly to people because most people understand that cancer is bad and that more research is needed. Despite that, when you're presenting your research, numbers are going to speak volumes, and scientists listening to your work are really going to want to hear statistics about how important this research is. So using something like this graph, which you've seen before, can be really impactful. This graph, like I said many times before, shows the cancer death rates for men in the US from 1930 to 2002. And the reason I really love this graph in trying to make a very strong and efficient point that there's a need for more chemotherapies and more treatments for cancer is that this graph is showing you that for most forms of cancer, at least in men in the US, the survival rates haven't really improved very much, except for lung cancer. And that doesn't really have much to do with finding good treatments for lung cancer. It's more of a lifestyle or behavioral change that has caused this decline in deaths in men. A lot of you mentioned this in your introduction section, and I really like this strategy of not only showing somebody that cancer research is needed, but giving them the reasons why. We need it not just because people are dying, but because the current strategies for developing chemotherapies are failing. And they're failing because the tumors themselves are becoming resistant to those chemotherapies, purely by chance. So you're all well aware at this point that it doesn't take more than one cell to become resistant by pure coincidence because of random mutations in that rapidly dividing cell to become resistant to one type of chemotherapy. And over time, if you keep treating that patient and you're not very successful, the tumor's getting bigger, the cancer cells continue dividing, and they continue to develop mutations that could be beneficial in terms of the cancer's survival. In describing chemotherapy resistance in cancer, the next thing that you want to give somebody is the answer. Why is your research the best research? What you're doing in the Discovery Lab is trying to identify a combinatorial therapy. You read about this in one of the Play Posit assignments. A combinatorial therapy takes two different strategies and uses them at the same time for two purposes. The first is to improve the efficacy of the treatment overall, and the second is to reduce toxicities. So in the case of a synergistic effect between a chemotherapy and radiation, if you're able to get a better effect by combining them than either of them individually if they were added, you'd be reducing toxicities because you could reduce the doses of both of those therapies. And that can be really powerful for somebody who has cancer. When you're describing your approach to someone, it'll be useful to explain to them how the individual treatments work. Now, in the case of your compound, you might not know. Some of the compounds that you're testing in Discovery Lab this semester are things that we don't know a lot about, but we have some clues about them working because of the data obtained in previous compound screens performed in the class. But with radiation, we do know how radiation works. Radiation causes DNA damage both directly and indirectly. It can directly cause double-strand and single-strand breaks, which leads to cell death, and it can cause indirect damage by splitting water and creating what's called free radicals or reactive oxygen species, which then can react with the DNA and cause double and single-strand breaks, again leading to cell death. One of the mistakes that people make when they're presenting their research is overestimating what their audience understands. You might be talking to a molecular biologist explaining your research, but the odds of them understanding how gamma radiation or ionizing radiation affects DNA is actually extremely low. So sometimes I know when I'm presenting my research, I feel like I'm spoon feeding people the details, and you really do need to do that. At the very least, a listener, even if they're an expert in molecular biology, will really appreciate the refresher to just remind them of how these things work. And also it helps get them on board with your research. If you're demonstrating credibility through knowledge of how your stuff works at the very beginning and how that leads to your research question, they're going to want to hear more. They're going to believe you. And one of the underappreciated things about science, especially now with social media and things being published on the internet before they're even peer-reviewed, is that you really need to be a great communicator. You need to be able to sell your work, like that elevator talk that we did in lecture a couple of weeks ago. So explaining things on a really basic level and then building up is going to go miles toward helping your audience understand what it is that you've done in this class. Finally, when you're explaining your research to someone, it's really important for them to understand not only what model system you're using, but why. We're using Drosophila Melanogaster, and we're using Drosophila Melanogaster because they're cheap, easy, and quick. Those are really powerful research qualities to have. They're easy to maintain and breed. We literally put them into a container that I bought at Target, and they're producing embryos that we can time and we can collect and make those embryos look like a tumor and find compounds that not only have the potential to be chemotherapies, but we've also identified compounds completely blindly that are chemotherapies. So this is a fantastic strategy for identifying things that translate well from your model organism, Drosophila melanogaster, to humans. Drosophila melanogaster have a very small number of chromosomes, but the number of genes represented that are mutated in humans that cause disease is 75%. And that's really powerful because if we look at human disease and look at all of the mutations that are causing human disease, 75% of them are expressed in Drosophila. So it makes it a really nicely translatable model. There are also lots of genetic tricks for generating and maintaining mutations. So a lot of mutations that exist in cancer would be developmentally lethal. In other words, if we tried to create a mouse that had a knockout of a gene that's common in cancer because we want to have a good model for tumor development later in life, a lot of the embryos where we mutate that gene will not survive. The same would be true of Drosophila, but we have some really amazing fancy genetic tricks to get Drosophila past development and then induce the mutation. So Drosophila melanogaster is a really great model for doing this type of screen. Please do practice the pronunciation of this. There's no better way to damage your credibility as a researcher if you can't say the name of your model system and your compound. So practice saying it. If you're not sure how to say it or how to say the name of your compound, let's talk about it and practice it because you don't want to be stumbling over that in your presentation during the symposium. So let's talk about drug development. How are drugs really developed and become what we see in the pharmacy? Historically, the context of this is you should understand that most of the drugs that are prescribed today were identified either from natural sources or were identified completely by accident. A great example of this is penicillin that was identified by Alexander Fleming. And the story behind that is really remarkable because penicillin was identified because of a really bad mistake in the lab. Alexander Fleming went on vacation and his graduate student left the plates that had bacteria that he was studying for other purposes in an incubator. And those plates grew mold, which is really common. Mold is the first thing that contaminates a bacterial plate. So Alexander Fleming came back and he identified, that half of his plates were moldy and he figured, oh my God, this huge experiment is totally ruined. But the genius of it was that he didn't just throw the plates away. He noticed that around the mold, there were areas where bacteria didn't grow. And he thought, that is interesting. There's something about the presence of the mold that's preventing bacteria from growing around the mold. And through a series of many more experiments, he was able to identify penicillin, which became an absolute game changer for human health. Now, another strategy that's been used to identify compounds, even chemotherapies, is to look at traditional remedies and to see what the active compounds are in those traditional remedies that historically have been shown to work but haven't been studied scientifically. And many of those, including colchicine that you use as a positive control, which is from a flower, were developed into drugs that we use today. A lot of you are using the same strategy in Discovery Lab this semester. There are people who are studying things like garlic, like venoms from animals. And one of the things that drives the entire National Cancer Institute's developmental therapeutics program is the cataloging of compounds that are present in natural products, in plants. And more recently, they're looking deep in the ocean at marine life to see if there are compounds there that might have some biological activity. Because the idea is that if you can fight fire with fire, you're going to have more success doing it that way than by trying to identify something brand new that's going to interact with an incredibly complex system. That said, a lot of the low-hanging fruit have already been identified, and I'll talk about that in just a second. So currently, the strategies involve identifying molecular pathways that maybe you could inhibit. We've talked about this too in terms of the hallmarks of cancer. An example of this is the upregulation of growth factor signaling, which is really common in a lot of forms of cancer. That when growth factor signaling through growth factor receptors is upregulated in a cancer cell, it creates a really great target to try to reduce the amount of growth within that cancer cell. Also, protein conformation is a newer way to try to think about how to identify compounds that might be chemotherapies or other drugs. Protein conformation refers to the shape of a protein. And we talked about earlier in the semester how mutations can cause a shape change that's very, very subtle. So can phosphorylation of a protein. And if you can identify the precise shape of a protein, you might be able to, using very sophisticated computer algorithms, identify a compound that could interact with a part of the protein and inhibit its activity. Finally, people are still considering the human genome. And if you're following this story at all, it's really interesting because an enormous amount of money was spent decades ago trying to sequence the entire human genome. And they yielded more than 30,000 genes with the hope that the identification of all of the genes in the human genome, they would be able to identify more drugs to treat human conditions. As of last year, the number of drugs identified from the human genome experiment has been extremely low. And some would argue that not a single drug has even been identified from the human genome. That said, gene sequence information can give us clues about the function of the protein. So if you sequence a tumor, you might then be able to get some information about what proteins might not be functioning properly in that tumor. So most drugs today were discovered just by pure chance. And recently, in the past, say, 20 years, pharmaceutical companies have been trying to use more sophisticated methods. The Discovery Lab approach is somewhere in between because we're doing a screening method, which means we're just testing like crazy, trying to identify something that will work, which almost makes it accidental. But the compounds that we're screening in, or the compounds that we're testing, we have information about based on molecular pathways, protein conformation, and gene sequence. So we have information about the proteins or the compounds that we're testing, but we're doing it in a way that makes it like we're just looking for something in a huge, huge number of compounds. Now, the second most important point that you should know about drug development is that bringing a new drug to the pharmacy is extremely expensive. We're talking about in the range of almost $1 billion for a single drug to become a drug that you find in the pharmacy. Overall, and these statistics are somewhat old at this point, 10 years ago, R&D, or research and development costs on pharmaceuticals, were exceeding $127 billion. And if you think about that, the number of new drugs per year is actually very, very low. So the rest of the money is being spent on drugs that never see the pharmacy because drug development is hard. So when we think about the cost of drug development, I think it's really useful to look to see what kinds of drugs are being developed. So I put together this chart that tracks by drug, which is product in the second column, the company that's developing it. Johnson & Johnson is J&J, indicated in the fourth row. The phase of clinical trial, the clinical trials have 4 phases. We're going to talk about that next week. The pharmacological class, or the type of protein that the drug is attempting to interact with, and the value of that drug today. So on average, a drug will cost $1 billion to go to market. But the top drugs, the ones that cost the most, are the ones that are costing anywhere between $3 and $5 billion. Why are they willing to put so much money into these drugs? The reason is longevity. And we'll talk about this next week when we talk about clinical trials. But when you're a pharmaceutical company and you're thinking about what disease you're going to go after, a curative drug is going to earn you less money. So these drugs that are listed here are almost entirely drugs that somebody would take for the rest of their life. So insulin is a great example. If somebody has diabetes, insulin is a Band-Aid. Insulin lets them eat and feed their cells and survive. But people who are insulin dependent take insulin every single day. So a company will be more willing to put a bunch of money into a compound that could be a drug that's taken chronically rather than something that's curative. The drugs that are the least expensive to develop are also the drugs that tend to be the ones that are easier to develop and may be curative. So an example of that is antibiotics. Antibiotics are curative drugs. If you have an infection and you go to the doctor, they give you a prescription for an antibiotic that you might take for 4 days or 2 weeks. You finish the prescription and you're cured. Antibiotics are cheap. So the problem with antibiotic development isn't that it's curative and isn't that it's cheap. The problem is that it's curative and it's cheap. In other words, a pharmaceutical company is going to be less likely to go after a compound that can only be taken for 7 days because they're not going to recover that $1 billion that it costs to take that drug all the way to the pharmacy. And we'll talk about the ethics of this next week, but you should know at this point that the cost of developing a drug is extremely expensive. Now, I mentioned that the total cost of drug development in the U.S. is very, very high, but it can't account for all of the drugs that make it to market. So where are those drugs that fail? If we look at a timeline of what happens with a compound, we can see that in the first phase, called drug discovery, there are about 5,000 to 10,000 compounds that you could put into this category. Of those 5,000 to 10,000 compounds that you're testing to figure out whether it works, to figure out which one works the best, and to figure out what targets those compounds inhibit or affect. Of those 5,000 to 10,000, you'll end up with about 250 that are looking pretty good. And you might then take those compounds and go into preclinical testing. Preclinical testing is before people, and this typically involves animals. So it's both in vitro testing that's more sophisticated, looking again at targets, and also in vivo testing, which is in life, typically in rodents. At that point, when you test all 250 of those compounds, you might end up with a very small handful that look promising. And then what it works out to be is about 5 statistically. So of the 250 that are looking good in animals, you might take the best of those and you'd end up with 5 that go into clinical trials. So you started out with almost 10,000 compounds and ended up with 5, because most of the compounds are failing, either because they're too toxic in animals, they don't work as you thought they would as soon as you move into animals, or they don't do anything at all, which is a common problem in clinical trials themselves, which again, we'll talk about next week. Of those 5 compounds that you're going to spend an enormous amount of money on getting in people and trying to test them to figure out whether it's toxic in people, if they can tolerate it, or if it actually works in people, only 1 new drug is going to result. So your odds of developing a compound that ends up in the pharmacy is 1 in about 10,000 compounds. Your experiments in Discovery Lab are down here in the drug discovery and preclinical phase. We're in an animal model in Drosophila Melanogaster. So we're bridging these two, because we're not going to be able to get precise target identification, but we're going to start gleaning information about what the compound does in doing the radiation, no radiation, or plus or minus drug experiment. We will not be moving these into clinical trials, but someone else might. After your information becomes public, after Tintin Sue's lab looks at your data, there might be a subset of compounds from our class that gets pursued. Now, we can look at this a little bit differently, where we have target selection, in vitro studies, in vivo studies, and then clinical trial and therapeutics, which again we'll do next week. But the progression typically is that you learn something about the research problem, which in this case is cancer. We learned about the hallmarks of cancer. We learned about the genetics of a cancer cell and how they develop mutations that might lead to drug resistance. And then what we do is we are in an animal model testing the compounds for efficacy, and then we'd be moving into a mammalian model. So we can think about drug development also in this way, where this is a typical progression of drug testing, where you might start with a target, which we kind of do, though our targets are kind of blinded to us. Then we go into in vitro studies, then in vivo, and then to clinical trials. This is the more typical progression of how things work right now. So let's use this as framework for thinking about how drugs get developed sort of in real life today. When we talk about drug or target selection, what we're talking about is trying to figure out something about the disease. So what is it about pancreatic islet cells that makes them not produce insulin anymore? What is it about your cancer cell that makes it really resistant to treatment? Most of this research takes place in academia, and by academia I mean at universities. It's considered basic research meaning it's in a lab and we're not treating any patients, but it's translational in the sense that the research can then move toward going into people. After target selection, you then would perform a series of experiments to identify a lead compound, which you read about, because you can't pursue every single compound. You have to identify one or two that you're going to put your money on, literally. Identifying compounds based on the target, or identifying compounds based on what we know about the diseased cell, for example, can be problematic. In theory, it's a really great approach, but in practice, you end up with a lot of compounds that can't enter a cell. So you might be trying to find a target by testing in vitro, where you'd have a purified protein to see if the compound can affect its activity. Or the compound can't be exported by the cell. And I know that we've been talking about multi-drug resistant pumps as being a problem in cancer, but cells stay healthy also by being able to move compounds out of the cell. In a cancer cell, we want to keep that compound inside to produce toxicity. But remember that chemotherapies almost always have access to every single cell. So we have to have a compound that can be exported by a healthy cell and hopefully isn't exported that efficiently in a cancer cell. After you identify a compound that works on a target, it's also possible that once you start testing in cells, there's an enzyme or something else in the cell that's actually going to destroy the compound. And this is something that's really hard to predict. Finally, after you have your compound that works fantastic on your target, a barrier to drug development is that it's toxic to mammals. So we might find compounds that work in Drosophila Melanogaster and look like they work really well in killing our tumor model, but as soon as we put it into a mouse, the mouse drops dead because the compound kills them. So targeted approaches are a really common, newer way to identify compounds that look promising as a pharmaceutical, but there are lots of barriers. It's a big hurdle to get past target identification and success in vitro and in vivo models. This is the biggest contributor to the failure rate from that 5 to 10,000 to 250 molecules. An alternative to targeted drug discovery is performing compound screens. This is a more traditional approach and has yielded a large number of compounds, but there's a very, very high fail rate in this also. But the good thing is that when you're developing compound screens, if you do it in the right model, you could identify compounds that get past that first fail, going from inhibition of activity of a purified protein to seeing how that compound actually works in the cell. In any case, even if you've identified a number of compounds using targeted development, or if you've performed a screen, you have to identify what you call a lead compound. So we're identifying hits from a screen, essentially. If we think about all of us as basically working as one big research group and testing a large number of compounds in our class, eventually we're going to end up with a very small number of compounds that actually work. Even though every single one of your projects has been informed by data, everybody in the class is testing a compound that either has published data showing that it looks promising or data from our own class showing that it's promising, the vast majority of your compounds are not going to kill the larvae. So from our large research effort as a class, we're going to identify hits, and those hits could become lead compounds. So this is where we are. We are identifying hits that we can then call lead compounds. Now, once the lead compound is identified, it's not enough. We need to make it even better. Just knowing that something works is great, but it's not enough to put all of our money on that one compound. What we're going to do, if we were a pharmaceutical company or if we were in a position to do more experiments aside from our basic experiment, we would start looking at the structure of our compound. Structures can be really complicated and they can be really simple. So if you haven't already, take a look at the structure of the compound that you're testing. We can get some clues about what your compound might be doing biologically by looking at the basic functional groups of that compound. And by functional groups, I mean just visually identifiable structures. So functional groups could be things like a circle. It could be something that has 6 carbons. It could be something that has 5 carbons. And what we see when we look at a lot of compounds that are drugs is we see themes of things like phenol groups showing up again and again and again, and specific shapes that make those compounds amenable to treating diseases in people. These shapes will define whether the compound is soluble, whether it can get into a cell, whether it will be degraded by a cell, can it be exported by the cell, and is it the right size to inhibit the function of something within the cell. Now you can take that, so say we look at the compound shape, the compound structure that you're studying. We might see that it has some interesting features. If we were developing your compound into a real drug, what we'd then do is we would try to change the shape of your compound to improve the properties of the compound. Because maybe your compound isn't totally soluble in water, and that's going to be really important. All of the compounds studied this semester are soluble in dimethyl sulfoxide. This is usually where we start with a solvent. Dimethyl sulfoxide is really great. It's used for things like patches. It will deliver compounds transdermally into your skin, and that's fantastic. You don't necessarily want to drink DMSO or inject it into your blood. So one quality of your compound that we would want to improve is its solubility in water, because that's the perfect drug. A drug that we can get into saline and inject into somebody's veins is a fantastic drug. And there are other properties that we can also improve on, like the ability of the compound to interact with proteins or not interact with, say, enzymes that degrade it. An example of medicinal chemistry that I like to use is codeine. So you've probably all taken codeine for a cold or a cough. This is the structure of codeine. And I want to show you how a very, very simple change in structure can lead to really dramatic functional changes of the compound. So here I'm showing you morphine and heroin. If you just glance at this, it's hard to see the difference among these 3 different compound structures. So we started with codeine, and I'm going to draw your attention to here. This is an oxygen hanging off of this big structure with a methyl group, which is a CH3 group. For our purposes, I'm not interested in you understanding what a methyl group is or a hydroxyl group or really any of the names of these structures, but I want you to understand that very, very subtle changes can change something like codeine from something that you take because you have a cold. This is actually over the counter in Europe. You can go and get ibuprofen that has codeine in it. It's fantastic. to morphine, which in no country can you get that over the counter. And the change is just in that same region where it takes out one carbon and two hydrogens and just leaves an OH group. So that very simple change changes codeine into morphine. And then if we add a couple of carbons or one additional carbon to the codeine, we end up with heroin. So this is a really tiny structural change, which can dramatically change codeine to morphine to heroin. Here's another example. You might be familiar with this one. So this is meth. This is meth that people are cooking in their kitchens and blowing up their houses for. Methamphetamine has a very simple structure. It has a carbon ring here, and it has two carbons hanging off with a nitrogen and a methyl group. So this is, in terms of compounds, you can see the difference between this and codeine. This is a very simple structure. And you might be familiar with this one already, but let me just erase what I've drawn here so you can really look carefully at this. Adderall, it turns out, is very, very similar to methamphetamine. And it's scary, right? Because people take Adderall all the time. In fact, in Boulder, pharmacies routinely every August run out of Adderall. They stock up and they run out because there's so many college students who are on Adderall. Adderall is a safe drug. Adderall can be used very productively for attention deficit. It also can be used productively to increase attention for studying, which a lot of people use it for that and recreationally. But it is a safe drug. Generally, Adderall is not killing people, but methamphetamine is, and the only difference is this tiny methyl group hanging off the side. To emphasize this even more, because I think that everybody now has heard about the drug called fentanyl, I want to show you that these doses here, so the amount of powder in the vials represent the amount of powder that would kill somebody. So if you took this much heroin, it would kill you. If you took this much fentanyl, it would kill you. And then this here is called carfentanil. It also has a very, very tiny structural change that changes fentanyl into a new compound. This tiny little dot will kill you. So if we're going from codeine to heroin, we'd make another small change to make fentanyl, and then we'd make another tiny, tiny change to make carfentanil. The deadliness or the ability, the lethality of these compounds increases incredibly. So medicinal chemistry is a really powerful approach for taking a compound that we know does something to making it do something even better. Sometimes when you make the changes, the effect is not the desired effect. Fentanyl, as it turns out, you might be looking at this and thinking, good God, why is this even a drug? Fentanyl is among a very small list of drugs listed on the World Health Organization as necessary drugs for any country. It can be absorbed through the skin, and it's an incredible painkiller. So it's used at times of war on the battlefields when people get injured. You can take a patch and put it on someone and in the field, in combat, make someone comfortable. But it has to be a medicinal dose developed by a pharmaceutical company, not adding fentanyl to something like pot that you would buy on the street. So you have to be really careful with these compounds, but if it's done in the right way, you can end up with compounds that have incredibly useful pharmacological properties to treat diseases. Now, after developing a target or performing a screen and having your lead compound, the next stage in drug development would be to do medicinal chemistry to improve the basic properties of that compound, and then to perform in vitro studies. And in vitro in our hands means cell culture. In other fields like chemistry, in a chemistry lab, if you're doing cell culture, they would call that in vivo because isolated cells for them are living. For us as molecular biologists, anything that we're doing in a tube or in a dish, we would call in vitro, even if it's a living cell. We don't move to in vivo until we're in an entire organism. The goals of performing in vitro studies after we have a lead compound that we've messed around with the structure and we're feeling really good about is to try to eliminate toxic compounds from our set, to determine the precise effect of the drug, to figure out the selectivity of the drug. In other words, if we're targeting, say, a specific receptor, we don't want it to target any other receptor. We also want to determine what the affinity of the drug is, or how high a concentration you need to get that drug to do its job. Finally, we want to figure out the mechanism of action. And we've been talking about this a little bit throughout the semester. Mechanism of action simply refers to how the drug is working. We want to understand how that compound is interacting with a cellular pathway, or interacting with DNA, or a specific protein. So sometimes this is abbreviated MOA. And you'll be talking about this on your poster when you get to that point after you have data. So at this stage, we're then taking our lead compound and we're making it even better, and we're learning more about it, which will strengthen our case when we go into in vivo studies where we end up spending a lot more money on experiments. The advantages of doing things in cell culture is that they can be done in a really short period of time. We don't need to follow an animal through an entire disease course. We can see what the compound is doing in a cell. We can also predict some of the toxic effects that we might see in the animals based on the toxicity that we see in the cells. Performing cell culture experiments or in vitro experiments greatly reduces the risk of clinical trials failing and of identifying toxicities much longer after a clinical trial is over. And finally, it's cheap. It's a great system in which to simplify a very complicated organism. If you can isolate the relevant cells first, we can learn a lot about what the compound is doing. Some problems with cell culture is that cells that are isolated in a dish are not an organism. Those cells don't have a liver that are going to process the compound before the compound ever gets to those cells in real life. We also can't predict how the compound is going to behave in other cell types or in the entire system. In other words, will the compound do something to your cells that are relevant to the disease, but also that are relevant to the system functioning as a whole? And this is also a problem with developing compounds, is just that cell culture is great, but in vivo or in life is much more complicated and harder to predict. The final stage before going to clinical trials is typically in vivo studies in animals. Now, I'm going to point this out next week, but I want to bring it up here. Clinical trials do not require animal studies. And we'll talk a little bit about PETA, the people who are interested in the ethical treatment of animals, because that's an important consideration when you're doing drug development, is the ethics of working with animals. And we're going to talk about that in 2 weeks' time. PETA will tell you that animal experiments are required for any development of any drug, and that's not true. But what is true is that to meet the criteria for going to clinical trials, it's extremely challenging to do it without testing in animals. Testing in animals is going to give us information about how the compound behaves in the organism as a whole. It'll tell us about how the compound is processed in the liver and how it makes it around the body. Does it stay the same compound when it reaches the tissues that you're interested in as it started out with? And the short answer is no, it almost never does. That's called the metabolic profile. Studying in animals will also give us an idea if there are things called off-target effects that could cause toxic issues, toxicity in the animals, and would also cause toxicity in people. And in animals, you can also do drug interaction experiments. This is very, very important, especially with people with chronic illnesses like cancer, because cancer patients almost are never on a single drug. Cancer patients may be on pain medications, they may be on heart medications, they might be on gastrointestinal medications. They're taking handfuls of medication every day. And understanding how your compound, which in the very next step of drug development is going to go into people, interacts with those compounds that are really commonly taken by the patients is very important in understanding how it's going to affect people taking the drug. Because you don't want to end up with a compound that's looking good in vitro, in vivo, in people, but then when we get to a large population of patients with the drug, with the disease, suddenly the drug fails. And you might not see that until phase 3 or phase 4, when hundreds of millions of dollars have been invested and people start dropping dead because they're just taking the regular medications that patients take with that disease. So drug interactions can be tested in animals and is an important component of in vitro studies. So we'll wrap up our discussion of drug development here, but remember that it doesn't stop here. You're the very first step in the preclinical phase of drug development. Information that you gain from your compound, which actually will be a significant amount of information at the end of the semester, will then be passed on to the next stage of drug development, which would be the in vivo testing. There would be extensive in vivo testing, probably for 3 or 4 years, before there's a possibility of a compound moving into people in clinical trials. And we'll pick up where we left off here next week when we talk about the whole process of clinical trials from phase 0, which is a new phase of clinical trials, all the way through phase 4, when compounds are already in the pharmacy. New compounds are still part of the clinical trial process when they're first released to patients. So we'll talk about that next week, and then we'll also talk about data reporting and bioethics, which is an important consideration in engaging in this type of research that I want to make sure all of you have a good handle on before you leave this class.